Welcome back to the deep dive. Today, we are undertaking a truly massive mission. We're plunging into what might be the single most comprehensive map ever created of the developing human immune system.
It's an incredible piece of work.
Specifically, we're focusing on that pivotal window, the second trimester, so roughly 13 to 28 weeks, which is really the period when the body sets the immune rules for life.
And it's the ultimate blueprint. For decades, developmental biology, it sort of operated under this assumption that the fetal immune system was, quiescent.
Quiescent as in asleep.
Exactly, asleep, or at the very least operating strictly to maintain tolerance so the fetus doesn't, provoke an inflammatory response, which would be catastrophic.
Yeah.
This study just fundamentally rewrites that rulebook.
And what a study it is. We are unpacking the insights from a landmark atlas published by he and their team. And they didn't just sample a few cells here and there. They mapped the entire architecture using some really state-of-the-art tech.
Oh, absolutely. They use single-cell RNA sequencing, or cRNA-seq.
Which is like reading the instruction manual for every single cell.
That's a great way to put it. And they also use scTCR-seq, which maps the unique receptors T cells use to identify targets. The scale is just staggering. They analyzed 2,868,420 high-quality immune cells.
Wow.
From 321 specimens across 23 different fetal and adult organs.
So the mission for us, for you listening, is clear. How does the developing fetus manage to build a fully functional, complex immune army, one that's capable of recognizing millions of future threats, while at the same time keeping that entire army strictly suppressed to ensure tolerance and survival?
That paradox is the absolute crux of the paper. They confirm the second trimester is this massive expansion phase. Immune lineages mature, they distribute, and they start to look strikingly similar to the adult landscape. And their key discoveries, they really fall into three essential areas. First, the surprising architecture that reveals widespread activity, which challenges that whole quiescence idea. Second, the precise molecular breaks that enforce immune tolerance. And third is something truly wild, the discovery of decentralized immune cell manufacturing throughout the body.
Okay, let's start with the architecture. If you put all these cells on a map, how different does the fetal profile actually look compared to an adult's?
Well, the initial data sorting, what's called principal component analysis, or PCA, it immediately separated the two groups. It wasn't even close.
A clean split.
A very clean split. The fetal immune system in that second trimester it's dominated by precursor cells, naive T cells, and these highly proliferative clusters.
Which makes total sense. It's still in the manufacturing phase. It's building the foundational pieces.
Exactly. It's the opposite of the adult system, which is enriched for the finished products, the memory and activated T cells.
Right.
But, and this is a huge but, within that manufacturing phase, they found a critical, a really unique intermediate population. And this is where we introduce the extrathymic precursor cells, or ET cells.
Yes, extrathymic. So outside the thymus. But isn't the thymus where T cells are supposed to go to school and mature?
It is. That's the traditional view. But they identified the specific subset of CD4 plus T cells, and you can spot them by the expression of a transcription factor called IKZF2. And crucially, they weren't just, passing through. These ET cells were broadly distributed across multiple organs way beyond the thymus itself in peripheral sites.
So if these T cell precursors are roaming around the body before they're fully trained, what on earth is their job?
They're the bridge. The researchers, they mapped the cellular lineage trajectory computationally, and they showed that these ET cells developed directly from the thymic precursor, or TP cells. But then these ET cells, they face a kind of developmental fork in the road. They can differentiate into one of two mature types.
Okay, what are those two fates?
Fate number one is your standard mature naive CD4 plus T cell. It expresses genes like STAT1 and KLF2 that basically tell it to stay quiet and rest.
The default path.
Kind of, but the more crucial fate number two, is their differentiation into regulatory T cells, TREGs.
And TREGs are the police force. They're the ones responsible for telling everyone else to calm down and tolerate our own body's cells.
They are the peacekeepers.
So why are these ET cells so important for generating them?
It all comes back to that specific gene, IKZF2. They found that high expression of IKZF2 biases the ET cells to preferentially go down that TREG pathway, to express FOXP3, which is the master switch for tolerance.
So it's like a dedicated factory for peacekeepers set up out in the field where they're needed instead of just back at headquarters.
That is the perfect analogy. It's a peripheral, highly efficient system to make sure the fetus generates the massive number of TREGs it needs to maintain tolerance everywhere.
So we have the peacekeepers being made efficiently. But now we hit what you call the memory paradox.
Mhm.
And this is the part that I found truly shocking. If the fetus is so focused on tolerance, why did the study find widespread evidence of activated and memory T cells across so many fetal organs?
That's the direct contradiction of the old quiescent model. They found T effector memory cells, effector T cells, even tissue-resident memory T cells. These aren't quiet, naive cells. These are cells that have seen something and are preparing for a rapid response.
But are they dangerous?
Yeah.
Are these fetal memory cells capable of the same destructive power as adult cells?
They're primed, but they're clearly operating with, let's say, restricted ammunition. For instance, a fetal activated CD8 plus T cell will express memory markers like GZMK, showing it's ready to remember. But if you look at the genes for its actual weapons, the cytotoxic arsenal like GZMH, GZMB, or PRF1, their expression levels are significantly lower than in adult cells.
So it's like they're building the weapons rack, but they haven't been given the explosive payload yet.
Precisely. Their potential for cytotoxicity is way, way down. They're learning and surveilling, but not really fighting.
Okay, so that brings us to the T cell receptor sequencing.
Yeah.
What are they learning about? Are they just seeing self-antigens, or are they seeing threats from the outside world?
Well, their clonal expansion, the ability to copy themselves rapidly once they see a threat is very low. We're talking less than 8% of T cells. In adults, that can be up to 80%.
So they're not overreacting.
Right. But, and here's the genuine surprise, the fetal samples contain a substantially higher proportion of TCRs that could specifically recognize a massive diversity of known antigens.
What kind of antigens are we talking about?
Everything. They found recognition signatures for self-antigens linked to things like lupus, common viral antigens like Epstein-Barr virus, and even microbes like M. tuberculosis.
Wait, how is that even possible?
It means the fetal immune system is building a massive library of potential threats incredibly early on. The priority seems to be breadth.
To sample the entire antigenic universe to establish a foundation.
Exactly. And what's really interesting is they saw that the frequency of these broadly specific TCRs actually decreased as development went on.
So the strategy is,
Yeah.
first, build a massive diverse library, and then later, after birth, you focus on expanding only the clones that encounter real active threats.
You've got it.
Okay, let's pause here and connect the dots for a second. We've established that the fetal immune system is anything but quiescent. It's complex, it's generating peacekeepers with these ET cells, it's establishing memory, and it's building this enormous library of antigen recognition. All of this should lead to massive inflammation, yet the fetus remains perfectly tolerant. How? This is the ultimate paradox.
It is. This level of complexity demands incredibly precise regulation. If T cells are active and they're seeing antigens, there have to be powerful, localized mechanisms stopping them from starting a war. And the study, it identified two primary molecular breaks that enforce this tolerance.
Let's talk about the first break. This one involves specialized neutrophils, and it sounds like some kind of anti-fuel mechanism.
That's a great way to think about it. This is where arginase 1, or ARG1, comes in. ARG1 is an enzyme that metabolizes L-arginine.
Okay, and L-arginine is what?
It's an amino acid that T cells absolutely rely on for proliferation and full activation. It's like high-octane fuel for them. Without it, the T cell just stalls out. And the key finding was that fetal neutrophils showed markedly higher ARG1 expression compared to adult neutrophils.
So the fetus deploys this army of arginine eaters to starve the T cells of their fuel.
Precisely. And these weren't mature neutrophils like we see fighting infections in adults. These ARG1 producers were in an early differentiation state, suggesting this suppressive job is baked into their fetal programming.
How did they prove this was the actual break in action?
They did a perfect experiment. They co-cultured fetal T cells with these fetal ARG1 producing neutrophils, and immediately the T cell production of inflammatory cytokines like TNF-alpha and IFN-gamma was just shut down.
Wow.
But here's the clincher. When they added an ARG1 inhibitor, or just flooded the culture with extra L-arginine, the T cells immediately woke up and started pumping out cytokines again.
So it's functionally proven that it's a brilliant mechanical way to enforce just steal the fuel. What about the second break?
The second break is a separate layer. It's a molecular signal. We're talking about the PTG-ES3, PTGR4 signaling axis.
Okay, break that down for us.
So they found that the molecular interaction between these two was way more frequent in the fetal immune environment. PTG-ES3 is an enzyme that produces something called prostaglandin E2, or PGE2.
And PGE2 is a bad guy, a good guy.
It's a globally recognized immunosuppressive molecule. Think of it as a universal do not activate signal. The fetal environment is just flooded with cells making it. And crucially, the fetal resting T cells, especially the CD8 plus ones, have a much higher proportion of the receptor for it, PTGR4.
So the environment is screaming, stand down, and the fetal T cells have their hearing aids turned all the way up.
A perfect system for maintaining homeostasis. And again, they proved it. They treated activated fetal T cells with a drug that mimics PGE2. Immediately, inflammatory markers plummeted. They added a drug to block the receptor and the inhibition vanished.
So you have two completely different breaks. One is metabolic, starving the cells of fuel, and the other is a chemical signal telling them to stay quiet.
That's the dual break system that explains how the memory paradox is managed.
It's fascinating, but before we move on,
Yeah.
let's just quickly revisit the tissue-resident memory T cells, the TRM cells. You said they were found across multiple organs, and that challenges the traditional view. How so?
Well, traditionally, we thought of TRM cells as local guards. They're stuck at one post, like the skin or the gut, ready for an immediate local threat, non-circulating.
Right.
But the TCR sequencing, it revealed extensive clone sharing. They found identical TRM clones, both CD4 plus and CD8 plus, in wildly disparate organs.
So the same cell clone was found in the intestines and the bladder.
And the trachea and the lymph nodes, exactly. This extensive sharing suggests they are not static guards at all. It implies a dynamic exchange, a migration system, even in the fetus.
Which means they're contributing to systemic immune surveillance, not just local protection.
It fundamentally changes our view of their mobility and their role in early life.
Okay, here's where it all comes together for me. We have active components that are being tightly regulated by these chemical and metabolic breaks, which leads to the final critical question. Where is all of this complex machinery being produced and distributed? This brings us to widespread hematopoiesis.
This is the third pillar of the study, and here we're focusing on hematopoietic stem cells, HSCs.
The mother cells for all immune cells.
That's right. You can identify them with markers like CD34 and a transcription factor called HLF.
And we've all been taught the timeline for where these are made, right? It starts in the yolk sac, moves to the liver and spleen, and then finally settles in the bone marrow.
That's the classic textbook model. But the atlas found these functional HLF-positive HSCs, well, everywhere.
Everywhere.
They were widely dispersed across multiple fetal organs that we previously considered non-hematopoietic. We are talking about the heart, even the skin, as early as 12 weeks.
Wait, the heart, as in the muscle that's pumping blood, is also making blood cells.
It is. It completely decentralizes the production model. It suggests a dynamic and spatially extensive manufacturing strategy during the second trimester.
And were they sure these cells were functional?
Oh, yeah. They took them out and did colony-forming unit assays, and they confirmed that HSCs from these non-canonical organs were fully capable of differentiating into multiple immune lineages.
But did the location matter? Did an HSC from the liver act differently from an HSC from the heart?
Yes, profoundly. This is the concept of organ-specific differentiation. They showed that the local microenvironment, the organ itself, actively influences what the stem cell becomes. So liver-derived HSCs preferentially generated myeloid lineages, monocytes, macrophages, neutrophils.
For first responders for infection? Makes sense for the liver.
Right. But the HSCs they found in the heart,
What about them?
they showed a dramatic skewing toward mast cells and erythroid cells.
That's an incredible connection. It suggests that the microenvironment of the heart is actively programming the immune cells it makes to serve its own local needs.
Precisely. The molecular reasons why are still a mystery, but the effect is undeniable. The body isn't just sending immune cells to organs. The organs are making the specific immune cells they need, tailored to their own function.
This atlas is. It's just going to be a treasure trove for research into developmental disorders.
Yeah.
But, as researchers ourselves, we should probably touch on the limitations the authors noted.
Right. It's important. While we have these amazing molecular profiles, a study like this, and cRNA-seq study, it can't fully capture the spatial organization, the physical interactions with the stromal cells, the actual tissue structure, which are probably what's programming those stem cells. That's the next frontier.
That was a truly rewarding deep dive. The idea that T cells are actively seeking antigens, establishing memories so early, but are simultaneously kept quiet by these potent, specific breaks. It just fundamentally changes how we view early human development.
What's so fascinating is realizing that the immune system's main job in the fetus isn't just defense. It's maintaining tolerance and weaving itself into the fabric of every developing organ. It's a partner in development.
So for you, the learner, here is a final provocative thought to chew on. If we know that hematopoietic stem cells found in non-canonical organs like the heart skew toward specific lineages, like mast and erythroid cells, does this suggest that early tissue microenvironments actively program the function of the immune cells they host? Or is it simply a matter of timing and developmental stage? And what implications might this have for treating tissue-specific autoimmune diseases later in life?
A big question to think about. Until next time, keep that curiosity flowing.